We will move to talk about the Leydig cells. As you know, we will find the Leydig cells in the interstitial space, so in the space between the um, seminiferous tubules, not inside the seminiferous tubules, but outside between the different seminiferous tubules, we will find the Leydig cells. They are polygonal cells. We can find them isolated or clustered around the blood and lymphatic capillaries. They can be isolated or clustered around the blood capillaries or the lymphatic vessels. Their nucleus contain many nucleoli, and their cytoplasm is granular, rich in smooth endoplasmic reticulum, mitochondria, and liposomes. What do those Leydig cells serve for? What is their function? In fact, their main action or main function is to synthesize the steroid hormones uh, and which are the testicular androgens. So they synthesize the androgens, they synthesize the testosterone necessary for many for the spermatogenesis inside of the seminiferous tubules. And also the testosterone is essential for the maintenance and functions of the sex accessory ducts and glands, and also it will stimulate the secretion of the sperm nutrients by the epididymis. So the testosterone is important for many functions in the male reproductive system. So those Leydig cells, they produce this testosterone, and they provide a complete steroidogenesis from the cholesterol provided as low density lipoprotein from the LDL or from the sorry, excuse me, they provide a complete steroidogenesis from the cholesterol or from the acetate. So they are able to synthesize the testosterone from the acetate or from the cholesterol as provided as LDL. Okay? And once they produce the testosterone, 95% of this testosterone is secreted in the plasma. 95% of the testosterone present in the plasma is coming from the Leydig cells, because we have also the adrenal gland which are on above the kidneys that produce also androgens and testosterone, but the major testosterone source of testosterone in the body are the Leydig cells. They produce 95% of the testosterone in the plasma that we find in the plasma. And also the testosterone can exert its activity directly as testosterone, or it will be converted or as or through its bioconversion to DHT, the dihydrotestosterone, which is more potent than the testosterone itself, or also it can be converted to estradiol, which is an estrogen, and that's how that's why it is important also, because it can act by itself, or it can be converted to DHT or to estradiol, and both of them have important actions in important functions in the male genital tract. So what are the roles of this testosterone? The testosterone have a role in the activation of the hypophyseal testicular axis, and we mean by this that it activates the crosstalk between the hypothalamus, hypophysis, and the testicles. And in fact, by the crosstalk, we mean the hormonal regulation between the hypothalamus, hypophysis, and the testicles, and that's what we call the hypophyseal testicular axis. It induces the differentiation of the male genital organs during the fetal period, and that's what we have seen earlier. that it induces the differentiation of the male genital organs into efferent ductals, epididymis, seminal vesicles, the ejaculatory duct. It will induce also the masculinization of the urogenital sinus and the external genitalia, and this happens also during the fetal period. And this is made by the under the action of the DHT, in fact, but the DHT is produced by the testosterone. So the testosterone also have a role in the masculinization of the urogenital sinus and the external genitalia. And also it is important for the acquisition and maintenance of the secondary sex characteristics, as the voice and hair, and also is important for the development of the male behavior, and the development of those secondary sex characteristics and male behavior starts from puberty. Because it's from puberty the gonads will start to produce the testosterone. And then in synergy with FSH, with the follicular stimulating hormone produced by the hypophysis, the testosterone ensures the maintenance and integrity of the germline, and the testosterone also contribute to the initiation and maintenance of the spermatogenesis. So it is important for the initiation of the spermatogenesis and for its maintenance. To keep the spermatogenesis occurring through the time, and also for the maintenance and the integrity of the germ of the cells of the germ cells. Now, the secretory activity of the testis is under the control of endocrine and paracrine factors or hormones. What are those endocrine and paracrine factors? So first of all, we will start to talk about the neuroendocrine control of the testicular functions. When we talk about a neuroendocrine control, we'll talk about the hypophyseal axis. In fact, it is the axis composed from the hypothalamus and the hypophysis. And this axis, or those two elements, which are the hypothalamus and the hypophysis, they constitute the major endocrine element, or the essential endocrine axis that will regulate the reproductive system through the production of endocrine hormones. So this hypothalamus-hypophyseal axis, by producing hormones, can regulate the reproductive system. We know that the hypothalamus contains neurosecretory cells. Those neurosecretory cells will produce GnRH, the gonadotropin releasing hormone. This GnRH produced by the neurosecretory cells will pass in the portal system present between the hypothalamus and the adenohypophysis. And in fact, if you remember, we said the earlier chapters that the portal system is a system of blood vessels between the hypothalamus and the adenohypophysis, which we call a portal system. Okay, so this GnRH will be produced and released in the portal system, the capillaries between the hypothalamus and the adenohypophysis, which constitute the portal system. So it will be released in the portal system, and in this way it will reach the adenohypophysis. And since the adenohypophysis has receptors for the GnRH, it can produce the FSH and LH under the action of the GnRH. Production of this FSH and LH by the anterior pituitary gland is pulsatile, which means it is regular or rhythmic at certain rhythm. Also, the production of GnRH is pulsatile also. So once the FSH and LH are produced under the action of GnRH, they will migrate in the blood circulation to reach the gonads, where in fact the FSH and LH will stimulate the gonads, and the gonads under the action of the FSH and LH, they can secrete several hormones as the testosterone and the estradiol, and they can produce cytokines as the activin and inhibin also. In fact, the testosterone is produced by the Leydig cells and the activin and inhibin by the Sertoli cells. The estradiol is mainly by the Leydig cells from testosterone. We have also the DHT, which is produced by the Sertoli cells from the testosterone. And then those produced hormones or factors can then exert negative feedback on the production of those FSH and LH, and on the production of GnRH. We have, for example, the testosterone and inhibit will exert a negative feedback on hormone production in the hypophysis and the hypothalamus. And in fact, the testosterone will exert a negative feedback on the production of GnRH by the hypothalamus and on the production of LH by the hypophysis, adenohypophysis, and the inhibin will exert a negative feedback on the production of the FSH by the anterior pituitary gland. As we are going to see here we have the hypothalamus that produces the GnRH, and under the action of GnRH, the adenohypophysis will produce the FSH and LH. Now, those FSH and LH will migrate in the blood circulation to reach the gonads. In the gonads, we have different types of cells. We have the Leydig cells, the Sertoli cells, the germ cells. Now, the germ cells, they don't have receptors to the LH and neither to the FSH, so they cannot be activated or stimulated by those two hormones. So, but in fact, the Leydig cells, they express receptors for the LH, and thus, under the action of LH, the Leydig cells can be stimulated to produce the testosterone. So, and in fact, to synthesize this testosterone from cholesterol or from acetate. So they can they produce the testosterone under the action of the LH. And once this testosterone is produced by those Leydig cells, in fact, a big part of this testosterone will pass in the blood circulation, and part of this testosterone will go to the inside of the seminiferous tubules to act on the Sertoli cells, because the Sertoli cells, in fact, they express receptors for the testosterone, so they can be stimulated by this testosterone. So the Sertoli cells, under the action of testosterone, they produce the ABP, which is the androgen binding protein, and this protein can bind the testosterone, and this way the testosterone can be concentrated in the testicular fluid and later in the plasma, in the testicular fluid produced by the Sertoli cells, and and thus by the it will contribute to the maintenance of a high concentration of androgens in the seminiferous tubules. In fact, this ABP will bind to the testosterone in the Sertoli cells, and not only the testosterone, the DHT also, because the Sertoli cells can convert the testosterone to DHT. So this ABP will bind the androgens in the Sertoli cells, and in this way the testosterone can be released in the lumen of the seminiferous tubule, and it can be concentrated in the lumen of the seminiferous tubule and later transported with the testicular fluid to the epididymis, where it can have important actions on the epididymis also. Yeah, in fact, it stimulates the secretion of nutrients for the sperm cells by the epididymis. Now, this is regarding the testosterone. So the testosterone is produced by the Leydig cells. It acts on the Sertoli cells, but not it cannot act on the germ cells because they don't have receptors for it. It acts only on the Sertoli cells inside of the seminiferous tubule. Now, this testosterone can exert a negative feedback on the hypothalamus by inhibiting the production of GnRH by it, and also it can exert a negative feedback on the anterior pituitary gland, where it can inhibit the production of LH by this anterior pituitary gland. Now, from the other side, the Sertoli cells, they have receptors for FSH, only for FSH and not for the LH. We have seen that the Leydig cells, they have receptors for the LH and not for the FSH. So the Leydig cells will respond only to the LH and not to the FSH. The opposite, the Sertoli cells express receptors for surface receptors for the FSH, and thus can respond to the action of the FSH. And but they don't have receptors for the LH, so they cannot be stimulated by the LH. So under the action of FSH, those Sertoli cells, they will produce also the ABP, the androgen binding proteins, which will bind to the androgens like the testosterone and the DHT, and by this they will allow the maintenance of a high concentration of androgens in the seminiferous tubules. And also under the action of FSH, the Sertoli cell will produce many messengers and growth factors and cytokines, which are important for the which exerts activities on the germ cells, on the spermatogonia, and they regulate the mitosis and the meiosis of the spermatogonia. One of those factors produced by the Sertoli cells under the action of FSH is the inhibin. In fact, the inhibin, as we have said earlier, it inhibits the mitosis of the proliferation of the spermatogonia. So this inhibin is produced by the Sertoli cells, but also it can exert a negative feedback on the anterior pituitary gland, and thus it can inhibit the production of FSH by the anterior pituitary gland. In summary, the Leydig cells, in response to LH, they produce testosterone, can produce it from acetate or from cholesterol. And this testosterone can either can go to the blood and lymph circulation, or it can do a local diffusion, the interstitial space between the Leydig cells and the seminiferous tubule. It can diffuse in the space to which means it will not go to the blood circulation, and it will exert a paracrine effect on the seminiferous tubule, and more specifically on the Sertoli cells. They act on the Sertoli cells, where they can induce the production of ABP by the Sertoli cells. And also those also they are important, those androgens. So the Sertoli cells, under the action of androgens, as the testosterone, they produce also factors which are necessary for the continuity of the meiosis and for the spermiogenesis inside of the seminiferous tube. And from another part, we have the Sertoli cells, which can respond to the FSH. They have receptors for the FSH and will respond to the FSH by producing ABP and inhibin. And they produce also the testicular fluid under the action of the FSH. In fact, the testicular fluid is produced by the Sertoli cells, and it will be secreted in this lumen, in the lumen. And this testicular fluid will carry, or will, by floating, by moving, it will transport with it the sperm cell from the seminiferous tubule to the epididymis. Also, we have said that the testosterone can exert a negative feedback on the hypothalamus and the anterior pituitary gland, where it can inhibit the production of GnRH and LH, and the inhibin produced by the Sertoli cells can exert a negative feedback on the production of FSH by the anterior pituitary gland. Now that we have seen the neuroendocrine control of the testicular function, or of the secretory activity of the testis, we will talk about the paracrine. We will move to talk about the paracrine control of the testicular function, or of the secretory activity of the testis. Now, this paracrine control is governed by the interaction, or is due to the is governed by the interaction of between the somatic cells, the somatic testicular cells from one side, and we have interactions between those somatic cells, paracrine interactions, and we have from another side interactions between those somatic cells and the germ cells. And just to remind you that the somatic cells that we will be talking about here are the Leydig cells, the Sertoli cells, and the peritubular cells. And we have three types of interactions. We have interactions between Sertoli cells and the Leydig cells. Okay, we have interactions between the Sertoli cells and the peritubular cells here in the lamina propria, and we have interactions between the Sertoli cells and the germ cells. We will start talking about the interactions between the Sertoli cells and the Leydig cells. Now, since the germ cells, they don't have receptors for the testosterone, and the Leydig cells produce testosterone, so we cannot have a direct interaction between the Leydig cells and the germ cells, because those germ cells, they don't have the receptors of the testosterone. However, we have the Sertoli cells, which have the receptors for the testosterone. And so by responding to the Leydig cells, by responding to the testosterone produced by the Leydig cells, the Sertoli cells will play a connection between the Leydig cells and the germ cells. So the Sertoli cells will serve as a connection between the Leydig cells and the germ cells, because they can respond to the testosterone. And in fact, the testosterone uh, um, under the action of the testosterone, the Sertoli cells can produce many many growth factors, cytokines that can modulate the germ cells divisions and functions. So that's how the Sertoli cells will be the connection between the Leydig cells and the germ cells. Uh, so the Leydig cells, by the testosterone, they control many functions in the Sertoli cells and also the germ cells. But at the same time, those Sertoli cells, they can modulate themselves the production of testosterone by the Leydig cells. In fact, we said that the Leydig cells produce testosterone that can act on the Sertoli cells. The Sertoli cells, from their side, they can have a paracrine action also over the Leydig cells by producing factors as the growth factors as the IGF-1, the insulin-like growth factor 1, and the inhibin. And both of them, they are factors that stimulate the steroidogenesis by the Leydig cells. When we say steroidogenesis, we mean the productions of steroids, the production of testosterone by the Leydig cells. And also the Sertoli cells can produce other factors like the TGF-beta and the activin, which exert a negative feedback or will inhibit the production of testosterone by the Leydig cells. So in this way, the Leydig cells will have a paracrine control on Sertoli cells through the testosterone, and from another side, the Sertoli cell produce factors that can either activate the testosterone production, or that what we call the steroidogenesis by the Leydig cells, or they can produce factors like the TGF-beta and activin, which will inhibit the production of testosterone by those Leydig cells. And as an example of how those factors can modulate the response to how can they modulate the production of testosterone by the Leydig cells. Here we have the example of the IGF-1, the insulin-like growth factor 1, which it has receptors on the Leydig cells. So once it is produced, this IGF-1 by the Sertoli cells, it will migrate through the interstitial space to reach the Sertoli cells. And since they have receptors on the Sertoli cells, they can activate functions, or they can exert their functions on the Leydig cells. And what will happen is that the IGF-1 will uh, increase the synthesis, will induce an increase of synthesis of the LH receptors inside of the Leydig cells. So in this way, since this Leydig cells will have an increased LH receptors, it can respond more to the action of the LH, and also the IGF-1 increase the synthesis of steroidogenesis enzymes. So they will increase the production, the synthesis of the enzymes involved in the production of testosterone. In this way, the Leydig cells become more responsive to the LH, and they will be able to produce more of the testosterone. Now we will move to talk about the control between, or the paracrine control or interactions between the Sertoli cells and the peritubular cells. In fact, the peritubular cells, they have androgen receptors. So they have receptors for the testosterone. So once this testosterone is produced by the Leydig cells, it can act also on the peritubular cells. And those peritubular cells, stimulated by the testosterone, under the action of testosterone, they can produce the PMOD S. This is a factor called the peritubular factor modulating Sertoli cell function. So we call it the PMOD S. So under the action of the testosterone, peritubular, sorry, the peritubular cells or the myoid cells will produce this P mode S factor, and this P mode S factor can act on the Sertoli cells in a paracrine way, so it will cross this basal lamina and act on the Sertoli cells. It will induce the production of the androgen binding protein, the transferrin and inhibin. So now we can see that the Sertoli cells stimulated by the PMOD S can produce the ABP. So we have seen earlier that the Sertoli cells under the action of testosterone they can produce this ABP, and under the action of FSH also, but also under the action of PMOD S they can produce the androgen binding protein, and they will produce the transferrin and inhibin also. Now, regarding the interactions, the paracrine interactions between the Sertoli cells and the germ cells, we have a reciprocal interactions between those two type of cells. Um, in fact, the germ cells can produce some factors, some growth factors like the EGF, the epithelial growth factor, which once produced by the germ cells can regulate the Sertolian function, so it can regulate many functions in the Sertoli cells. And also the Sertoli cells, as we have seen earlier, they produce substances involved in the metabolism and the regulation of the germ cells division. So the Sertoli cells produce cytokines, growth factors, metabolites responsible for the germ cells division and meiosis, and also for their they serve as a nutritive support for those germ cells by the substances and metabolites that they will produce. And also those Sertoli cells, you know that they phagocytose the residual bodies formed or released during the maturation of the sperm cells. Now, as a result of this phagocytosis or this phagocytosis of the residual body, will induce the production of interleukin 1 and interleukin 6 in the Sertoli cells, and as a result of the production of those interleukin 1 and interleukin 6 by the Sertoli cells, in fact, they will be produced by the Sertoli cells, and they can act on the germ cells where they can control the DNA replication, the mitotic and before the meiosis, the replication of the DNA before the mitosis and before the meiosis. So the phagocytosis of the residual bodies produced during the spermatid's maturation into sperm cells will trigger the production of interleukin 1 and 6 by the Sertoli cells. So those cells will produce the interleukin 1 and 6, and those interleukin 1 and 6 will be secreted by the Sertoli cells, and they will act on the germ cells in a paracrine manner, where they will control the DNA replication of spermatogonia and primary spermatocytes before they start the meiosis. So here we will see a summary of the paracrine and endocrine interactions that we have talked about. In fact, the GnRH, the hypothalamus produces the GnRH, which will act on the anterior hypophysis and will induce the production of LH and FSH. The LH, since it has receptors on the Leydig cells, will act on the Leydig cells. Those are the Leydig cells, and will stimulate the production of testosterone by the Leydig cells. So here we have the testosterone produced from the cholesterol in the Leydig cells under the action of the LH. This testosterone has receptors on the peritubular cells and on the Sertoli cells, so it can act on both of them. When the testosterone acts in a paracrine way on the peritubular cells, it induces the production of the PMOD S factor, and this factor its turn, this factor will can have a paracrine action over the Sertoli cells, so it will induce the production of inhibin in those Sertoli cells. And from another side, this testosterone can directly act on the Sertoli cells, and this testosterone will modulate many functions in the Sertoli cells and will induce the production of cytokines and factors by those Sertoli cells that will have effects on the germ cells divisions and meiosis. Now, now, as we said, those Sertoli cells, they can produce inhibin, and this inhibin can exert a negative, can exert a positive feedback on the production of testosterone by the Leydig cells. So they stimulate the steroidogenesis by the Leydig cells. We said also that the Sertoli cells can produce the IGF-1 also, which have a positive effect on the production of testosterone by those Leydig cells. Now, under the action of FSH, we said that the FSH is also produced by the anterior hypophysis under the action of GnRH, and since the Sertoli cells, they have receptors for this FSH, so the FSH can stimulate in the Sertoli cells the production of inhibin and activin, where the inhibin can exert negative feedback on the production of FSH by the anterior pituitary gland, and the activin at the opposite of the inhibin will exert a positive feedback on the production of FSH by the anterior pituitary gland. We said also that the inhibin and activin, they have an action. The inhibin will inhibit the proliferation of the spermatogonia, and the activin, when they are present, they induce, or they have a positive action on the proliferation of the spermatogonial cells. And also, as you can see, this activin can induce the transformation of the testosterone to estradiol in the Sertoli cells. However, the inhibin will inhibit this transformation. So this is all regarding this neuroendocrine and paracrine control between the different cells of the seminiferous tubules and of the testicles, and also the neuroendocrine control with the hypothalamus hypophyseal axis.